Today we want to see the practical internal structure, primary structure of monocot and dicot. Prior to it, we should be know how the section should be taken here. So we firstly we see the video. Okay. So these are the requirement here. So we required the sample of monocot and dicot stem. For the monocot, we select the mage stem. For the dicot, we select the sunflower stem. Because why we select this one? It is easily available, as well as if you see the both mage and sunflower, it is a very soft as compared to the other stem. Next, we required the sharp blade. Next, the watch glass, slide, cover slip, brush, glycerin, and saffronic. So here we see here. So these are the monocot stem. This is the dicot stem. These are the requirement here. Next we see the technique here. How we can uh, hold this stem on in between the finger, and how we are get this section. See here in the video here. So this is the procedure here. So this is the way here. In between the two finger, we uh, hold this section, and through this another hand, we can hold this blade, and this is the procedure here. So this is the technique that is called as this skill here. So we are not get the very thin section within the 2 or 3 sections. If you get the continuous section, then automatically we have get the very thin section, and that we want to require here. So after taking the section, we have the collected sections are put on a water watch glass with having the water, and those section we have that float on the water that we have selected here, and then we have placed on a cover, that is a slide. See, that's a floating section here. जेवढा thin section असेल तेवढा तो float होईल. This is the stain here. Actually, in past practical, we see here we required the saffronin. So this is the saffronin here. 1 to 2 drop saffronin we have added. That saffronin get diluted, and then we can select that section. There's a two method here. We directly added this saffronin in the watch glass, which contained in the section. Second one, where we get the slide, put the section on the slide, put 1 to 2 drops of water, and then we added the 1 drop of that is the saffron. So this is the different way here. We want to easy, we can select this one. This is the next method here. That whatever this extra staining takes place, that should be removed. So with the help of this water, we have put transfer this section until the next year. Okay. After the washing, after the removing of the extra stain, this section put into the on the slide here in the center region here. So if you see here, so this is the method here. After then, we can added the glycerin. So why we use added the glycerin? If you see at the time of a practical, we use the only water. That water get evaporated. Okay, and due to the evaporations, the section get some kinds of a shrinking, and we cannot be see that's a good section here, or we can observe under the microscope. We cannot be observe detailed structure here because the section gets shrink and it become a dry. So for avoiding the shrinking, we add the glycerin here. Okay, after adding the glycerin, we can put the cover slip. So this is the way here. Single drop of glycerin here added, and then we put the cover slip gently to avoid the formation of bubble. We should be remove the bubble with the help of water or some kinds of a glycerin here. So next section, we want to examine the dicot or monocot step after the preparation of the TS of the slide. Then we have put this slide under the microscope. We adjust the eyepiece 10x or 40x, whatever be, and then we can watch it. So this is the methods here. After this sectioning, we observe the my that's procedure here. Then we see the next part here. So we see the detailed structure of internal structure of monocot, or you call it the anatomy of monocot. So generally we see first steps is the procedure, how we have get this section. After that, we can watch this section under the microscope. First we see the monocot stem. So here, this is the objective. First, we should be understand the internal feature of monocot. So in which here we want to study the tissue differentiation in monocot. First, there is a different kinds of a tissue system. First is the epidermal tissue system. Next is the ground tissue, and next is the vascular tissue. Okay. After then, if you get the idea about what is the tissue system, how it get differentiated. In the monocot, there is a typical tissue system arrangement. In dicot, we have get the typical system arrangement. So after then, we have get a differentiated. In monocot, we have the typical tissue. In the dicot, we have the typical tissue system. So we see one by one. So internal component of monocot stem. So in this stem, they have the four types of a tissue. First, we have the dermal tissue. Next is the hypodermal tissue system. Next is the ground tissue system, and last one is the vascular tissue system. So if you see in the section here, when we observe under the microscope, we can see these kinds of organization. The first layer that we call as a dermal tissue system, especially they have made up of the epidermis. After this epidermis, there is the presence of a second layer that is called as a hypodermis below the epidermis. Yeah, especially depending upon the plant, we can find out the single layer or multi-layer of epidermis system. Most of the grasses they have the single layer, but if you see the study of the xerophytic, these epidermis are the multi-layer. After the epidermis, there is a several layer of a hypodermis. If you see here, this is the thick cell region. After that, the hypodermis, the next cell that is called as a ground tissue here, and in the ground tissue, we can find out the vascular bundles here. These are the vascular bundle. So when we have observed the monocot and dicot, where there is a main difference we can find out in the dicot, the uh, ground tissue are present into the cortex region, but in the monocot, this cortex region is absent. If you see here, cortex region is totally absent. Directly you can find out epidermis, hypodermis, ground tissue, and vascular bundle. So this is the main difference here. This is the first point: is the cortex region is absent in a monocot. Here, next if you see dermal tissue system, one by one we will see. It is a composed of epidermis. So if you see, this is the some kinds of a first layer. These are the elongated, packedly arranged cell. So this is the barrel shaped cells we can find out here. After mostly this is the layer, single layer. This tissue is made up of parenchymatous. Cells are compactly packed. Next property is these are the thick layer cuticle. Along this epidermis, there is some kinds of a waxy coating are present. This is nothing but we call as a cuticle here. some of this, if you see, especially in this mage, this cell wall is the silica fight. That means silica have get deposit inside the epidermis cells. Epidermis persistent along as a stem, so there is a no secondary growth we can observe here. In some plants, there is a presence of hair-like outgrowths that is called as a trichome, but in these sections we cannot be observed here. So there is nothing, any kinds of a hair-like structures we can find out here. So
सर
trichomes.
तुमचा आवाज नीट नाही येते.
सर आवाज येत नाही.
आता आता येतोय का? Hello.
हो.
Okay, so these are the dermal tissue system here.
येतोय सर.
So here we can find out this epidermis characteristics. First, dermal tissue system composed of epidermis. Second one is the single layer made up of parenchymatous, or epidermal cells are compactly arranged. This epidermis having the thick layer, cuticle layer. This cell wall is the silicafied, and most next is the epidermis persists of a long persist as long as the stem. So here is no secondary growth we can find out. And last one is the some of the member or some of the plant there is the
सर
absence of.
आवाज नीट नाही येते.
1 minute. Hello, आता येतो का आवाज?
Yes sir. हो सर.
Okay, so these are the dermal tissue system. So if you see on the dermal tissue, this tissue system is interrupted by a particular epidermal cells that nothing but we call as the stomata. So if you see, observe, इथे ऑब्झर्व होते बघा, ही epidermis इथे interrupt झालेली आहे. So this we can, on this epidermis we can find out the stomata also. Next, if you see what is the function of this epidermis here. So first limiting layer, or we call as this is the outer boundary. It have get a protection. This we call as we call as a dermal layer, or we call as a first layer. Sometimes they have some kinds of a silica depositions. It's provide the mechanical support. Next, on this epidermis there is a presence of some kinds of a waxy coating that is called as a cuticle. It's prevent the transpiration. And next is the this interrupted epidermal cell that is called as the stomata. It's allowed the gases that is called as the exchange of gases as well as the transpiration. Okay, so these are the main function of this dermal tissue system. Next, if we see second layer that is called as a hypodermis. So below the epidermis, the hypodermis are present immediately. If we see here, this is the single layer, and this is the reddish 1, 2, 3 up to the 4 layer. We can find out this hypodermis here. This is also, if you see, this is the some epidermis and hypodermis having the typical structure here. Epidermis having the some kinds of elongated, but if you say hypodermis, it's having the polygonal structure here, and it's also having the compactly arrangement. This hypodermis is made up of multi-layer and sclerenchymatous tissue. Again here, alternately we can find out some kinds of chlorenchymatous patches because of due to the presence of a chlorophyll pigment. Below the cholenchyma, hypodermis is continue. Here we can observe some kinds of vascular bundles embedded in this vascular in the hypodermis. So if you see here, in between the hypodermis, these are the some kinds of a group of tissue. This is nothing but we call as a vascular bundle. So what is the function of this hypodermis? It's provide the support and protection. mechanical supports. It's become a strengthening to the some kinds of a to the plants here. Next, if we see the ground tissue, so in between these are all this ground tissue. So below this here hypodermis, this irregular circular, there is the presence of some kinds of a gap here. This region we call as a ground tissue system here. So here we cannot be seeing distinctness. Yeah, distinct. जसं आपल्याला डायकॉट मध्ये कॉर्टेक्स रिजन वेगळा सापडतो, तसं इथं आपल्याला वेगळा सापडत नाही. म्हणून ground tissue is not differentiated, composed of a cortex only. If you see this, this continue परत जर मीटिंग संपल्यानंतर परत लॉग इन करा सेम लिंकला. This cortex especially is made up of a parenchymatous cells. The cells are large, circular in nature, loosely packed here. Loosely packed means we can find out this cap. इथं दिसतंय बघा आपल्याला. इथं प्रत्येक सेल मध्ये आपल्याला irregular structure असल्यामुळे इथं गॅप दिसतो. So we can find out some kinds of a loosely packed with having the intercellular space. That is called as a intercellular space. So in aquatic monocot, if you see some of the member of this monocot, we can find out the air-like chamber that is called as a parenchyma. In most of the plants, the center portion is hollow. If you see in this Triticum aestivum oryza, आपण जर गहू आणि तांदूळ जे सेक्शन घेतला तर आपला मधला इन मिडल रिजन आहे हा मोकळा पोकळ असा पडतो. ओके, सो डिपेंडिंग अपॉन दी प्लांट, आपण प्लांट कोणतं सिलेक्ट करतो त्याच्यावर. तुम्हाला जिएमएज मध्ये हे होलो स्टेम सापडणार नाही, पण गहू बाजरी याच्यामध्ये गहू आणि तांदूळ मध्ये त्याच्यामध्ये तुम्हाला मोकस स्टेम सापडेल. ओके, what is the function of the ground tissue? So it is used for the storage and bearing the vascular bundle. Storage जे काय स्टार्च तयार होतं, जे फूड मटेरियल तयार होतं, ते फूड सर्व ह्या ग्राउंड टिश्यू मध्ये स्टोर केलं जातं. म्हणून ज्या सेल्स बघितल्या या लार्ज आणि इरेग्युलर मध्ये. ओके, सो नेक्स्ट वन, the tissue system that is called as the vascular tissue system. So especially this composed of a vascular bundles. That is a vascular bundles. There is a group of cells we can find out here. So if you see, this is the bunch. आपल्याला इथं bunch दिसतंय बघा. असे दोन लार्ज सेल दिसताय आणि त्याच्यानंतर परत असा एक आहे. पण इथे तुम्हाला लगेच डिस्टिंग्विश होतंय की हा एक वेगळ्या टाइपचा टिश्यू आहे. So these are all are we call as the vascular bundles here. So if you see in the monocot, the third one differentiated is all these vascular bundles are scattered arrangement in the cortex. This is the important differentiated, because through this vascular bundle we can classify the monocot and dicot stem. Okay, and here you can find out vascular bundles are numerous. Many vascular bundles we can find out in the ground tissue, and this ground tissue are this scattered. Anywhere we can find out this ground tissue. In dicot we cannot find it scattered. The vascular bundles are in the form of a ring here. Okay, so next point, if you see vascular bundles VB are widely separated from each other. If you see here, one place here, second place here, third place here. In here also you can find out there is a much more gap you can find out here. So there is a widely, that is called as a widely separated here. Next, if you see, the size also get varies. Vascular bundles also shows the some kinds of a different size differentiation. Vascular bundles in the outer regions are smaller, while inner regions is the larger. If you see here, this one, because this is the early cells here, this is the mature cells. Early cells having this some kinds of a smaller vascular bundle, but mature regions there is a we can find out the larger vascular bundles. If you see in the figure, we can get differentiated easily. And in this monocot, we can find out vascular bundles are cojoint, collateral, and closed. Cojoint, that means there is a, these are the in both these sides you can find out the particular kinds of xylem tissue, and these are the collateral to each other, and closed means what? There is nothing, any kinds of a cells find out in between the vascular bundles. And so we call as a closed. Otherwise, we call as a, if there is a presence of cambium ring, then we call as a open. ओके, इथं कॅम्बियम रिंग ऍबसेंट आहे आणि इथं कोणतंही सेल्स नसल्यामुळे आपण त्याला काय म्हणतो? क्लोज्ड. ओके, आता इथं बघा कोलॅटरल म्हणजे दोन्ही साइडना सेल्स अरेंजमेंट आहे. ही तिथे सेंट्रल रिजन आहे आणि हा क्लोज आहे. सो दिस इज द differentiation or characteristics of a vascular bundle of monocot. I again repeat it. It is a composed of a vascular bundle. It is a numerous in number, scattered arrangement in the cortex regions, widely separated from each other. It shows a very size differentiation. Outer region is having the smaller, central region is having the larger, and vascular bundles are cojoint, collateral, and closed type of vascular bundle. So this is the vascular bundles. If you see here again, this is the next characteristics here. In between this, actually the vascular bundle is made up of a particular kinds of cells. These cells we call as a xylem and phloem cells. Another one is the cambium cells, but cambium is absent here. Okay, so here in this monocot, cambium is absent. So there is a, we call as a closed vascular bundle. Vascular bundle composed of xylem and phloem. If we see here, these are the phloem cells, and if you see these larger cells, we call as some kinds of a xylem. In the xylems also we can find out the two types of xylem. One we call as a protoxylem, next we call as a metaxylem. Protoxylem are the earlier young cells, metaxylems are the mature cells. So if you can see here again differentiation, and these are lie bilateral side of the vascular bundle. So we call as a collateral closed type of a xylems, vascular bundle. Okay, and next if you saw enlarged, xylem is a enlarged type. Enlarged, enlarged means inner side the xylem are arranged, so inner side of a ear. Okay, next if you see protoxylems arranged towards the interior, while the metaxylems arranged towards the exterior. So inner side we can find out the some kinds of protoxylem, and outer sides we can find out the metaxylem. Xylem elements are arranged in a V shape. कसं आहे ते? V shape. तुम्हाला इथे दिसतंय बघा. So this is the typical arrangement, V shape. इथं metaxylem आहे, ही protoxylem आहे inner side ला. So it's form of V shape. Metaxylem elements are at the form arms of the V. ही बघा इथं metaxylems इथे. हे काय? V च्या arms आहे. Okay. And protoxylems occupy at the junction of V. इथं हे junction आहे. म्हणजे हे arm इथं join करतं म्हणून हे V shape. Okay. So this is the particular arrangement of the monocot anatomy. So here, if you see again, if you again, it is sufficient for FYBSc, but if you enlarge it, we go. So here, so we separate xylem. Metaxylem composed of two large vessels here. Metaxylem vessels are pitched. Metaxylem tracheids are between the metaxylem. So if you see here, xylem cells are made up of different kinds of cells here. Actually, xylem we call as a tissue. Xylem we call as a tissue. This tissue is made up of different kinds of cells. So if you see here, vessels, tracheids, okay, parenchyma cells, cholenchyma cells. These are the composition of this xylem tissue here. So protoxylem composed of a few vessels. Next, if you see, protoxylems are annual and springly thickening. Protoxylems elements fuse to form a lysogenic activity. This activity is called as a protoxylem lacuna. This if you see in this asparagus, शतावरीमध्ये आपल्याला तसं दिसतं. If you see next here, phloem tissue situated outer to the xylem. These are the phloem tissue. आणि ह्या आउटर्स म्हणजे आतमध्ये झायलेम टिश्यू आहे आणि बाहेर काय फ्लोएम टिश्यू, म्हणून त्याला एक्झार्ज, म्हणून त्याला काय म्हणलं एक्झार्ज टाइप ऑफ व्हॅस्क्युलर बंडल्स. दिस इज द रिजन या. दॅट मीन्स इनर साइडला झायलेम आणि बाहेर साइडला फ्लोएम आहे. Next, phloems composed of sieve tubes and Campanian cells. This is the component of the phloem tissues. First, we are having the sieve tubes, Campanian cells, phloem parenchyma. Vascular bundles, most of cases are surrounded by the bundle seeds. So these are the bundle seed cell. These cells, bundle seed cells, are made up of sclerenchymatous tissue. So if you see differentiation, in dicot, epidermis hairs are present, that is called as the trichomes. In the monocot, epidermal cells are absent. Hypodermis sclerenchymatous, hypodermis sclerenchymatous, endodermis present in a dicot while it's absent in a monocot. Ground tissue